Yeah, so to get started, um, my name is Tim Monko. I'm a postdoc at the University of Minnesota, and I have a background in neuroscience, and currently I'm studying embryonic brain development and iron deficiency. But I have a big interest in bioimage analysis, sort of fell in love with that in graduate school and want to keep pursuing it. So it's wonderful that you're all here. This is kind of my opener slide. I presented this at Bioimaging North America, which if you are, well, probably anywhere in the world, it's my favorite conference that I've ever been to. So please uh, become involved in BINA and go next year. It's in Montreal. It was awesome. And so I was able to present this slide as a flash talk. And I titled it High Throughput Image Processing with napari-ndev for the generalizable quantification of neurodevelopment, but really you can use this tool for anything. So what I'm presenting to you today is a plugin that I've been developing in Napari. If you're unfamiliar with Napari, it's sort of like ImageJ, open source, but in Python. And I fell in love with it. So to kind of give an overview before we get going, I specifically designed napari-ndev to address the gap that I saw 2 years ago, at least, between the Napari viewer, which has so many great plugins and tools to manage images, and then kind of that batch processing, very practical side. I have all these images from my microscope. I want to get data. How can I do that efficiently without writing code? Because code is a big barrier for people when it comes to bioimage analysis. So a few kind of goals formed around napari-ndev or NeuralDev when I was designing it. I really wanted it to accept diverse image formats and maintain important metadata like scale or channel names. The reason for accepting diverse image formats is that one of the most tedious things when it comes to coding is trying to kind of get your images into a format that can be used by a particular plugin. So if you have all these different microscope formats, napari-ndev can kind of handle them into the same format and use them throughout the pipeline, which is a super great feature. It also allows the use of what are called advanced workflows. Well, and for me, so a tool that I'll show you today is Robert Haas's and Talia Lambert's Napari Assistant, which allows you to kind of do live image processing in the Napari viewer. And then we take the output of this called a workflow and allow it to be shared. So if we look at the middle here, we can see just how easy batch processing is, where we select a directory that contains our images of interest, a result directory, and then a workflow file that we've designed in the Napari viewer by hand if we want. And then we just select the tasks that we want to do. It will also default to certain tasks, but you can select more. And you just hit batch workflow, and this will run and output those images that you desire. A super great tool. We also have the capability in napari-ndev for really user-friendly sparse annotation for batch training machine learning classifiers. You can see, for example, in the top right in this logo, this is looking at multiple different features of a developing neuron. And these morphologies of these complex cells are traditionally very hard to segment with intensity-based processing, and deep learning tools have not really kind of bridged this gap in this complex, detailed morphology. And so machine learning, as you can see, gets us really close to absolutely excellent results in complex segmentation. And so one of the priorities of napari-ndev is to really make machine learning super usable for a novice. And finally, I think a key here also is that we need to measure our data, right? We need to, for whatever reason, do statistics, make comparisons, evaluate the quality of our data. And so a big core element of this is that we have flexible data measurements, and we can then summarize that data so that you can use it in a format that you might be familiar with, like Prism or Excel, rather than having this, spreadsheet with millions of rows and being uncomfortable and not used to that. And that typically requires coding. So you can see a few different applications here on the right. We've got kind of this logo with, the machine learning segmentation, and then intracellular proteins of interest that we want to quantify. We've got nuclei segmentation, which we'll actually work on in the tutorial, and we have a specific region of interest that we're specifically wondering how many cells are in this area, because in this embryonic brain, this will become a certain region in the future brain, and so we can consistently annotate our images. This is something that's currently difficult to do in Napari in my experience. Am I not sharing? Yeah, Dean, thank you for your question. Can you please share the presentation? Um, yeah, actually, just let me do that quick. I will share, um, and I will just make this, anyone with the link can view it. And I will just put it in here if you'd like to look at it yourself. Everyone in the meeting. And then I'll try to also upload this to the documentation afterwards. So please let me know if that works, Dean. Yeah, so that's kind of the overview. So before I get started more so, I want to kind of acknowledge the people that have supported this work. My advisor is excellent. Tommy Bastian has just sort of supported me to run with this for the past 2 years. And then a few particular people have been kind of the main troubleshooters to this project. Emma and Jane from our lab, Elizabeth and Joe Chen's lab, and Chloe in Helen Vuong's lab. And these are both undergraduates and graduate students. A big priority of mine is that a brand new undergraduate can sit down at a computer and do image analysis for the first time in their life within an hour and get the data output. So today I hope to show you a tool that I hope can do that for you and the collaborators and trainees in your lab. So well, I'm going to do like a little survey. We're going to try it out in something called Slido, which I thought was cool at Bina. And I want to make sure that everyone's set up for this. I'll then discuss a little bit more about the origin and goals of napari-ndev. And then we'll have interactive tutorials. This is honestly my first ever workshop. I'm very excited. I'm also very nervous that this will be challenging, especially in a virtual environment. And so I would love your feedback in particular if you want me to kind of do the walkthrough. There's materials, then you can kind of go through the documentation. I think it would be great if you tried to do it yourself. That's kind of my goal. And then I'll be here at any moment for any questions, kind of like a big virtual meeting room. So maybe it'll be awkward and quiet at times. But that's my goal. And then maybe if we have more time, we can talk about maybe usage that you might have or features that you would be interested in. And then I have maybe take a survey. I haven't designed a survey, but I would love the possibility of making a survey. So maybe you'll even just make a Google survey link that you can share your email, and then later I can email you a survey so that you can give feedback. And again, as I said before, please interrupt this workshop at any time to ask questions. So hopefully now you can kind of see this screen that's showing a Slido. And if you take your phone or go to slido.com and enter this number, you should be able to answer this poll. So I've kind of 3 questions because I want to kind of judge where the audience is coming from. And yeah, it'll show up live. So I'll just wait a few minutes to get a few responses. Wow, this is great to see how many people feel comfortable. I think I'll try to adjust this to make sure that I don't over-explain some just kind of basics of image analysis. So thank you all for coming. I hope you find this useful. So this is great to see. I think if I just click, it'll go to the next slide. There we go. So this asks, how experienced are you with Napari? And I'm kind of interested, like, is it your first time with Napari? Have you mostly used ImageJ or Cell Profiler or some other kind of QPath proprietary tool? Um, or if you're experienced, and that'll partly change how I uh, explain um, how to even download it if you haven't gotten started. Okay, this is good to see. So a lot of people are new to Napari. Wonderful. I hope that this is a good introduction. There are so many great tutorials on Napari. I hope I can kind of live up to that in any way. So thank you all for your responses. This is fantastic. Okay. So then the last question, if you could kind of organize some of these things, even if you didn't know about them to begin with, you should be able to kind of slide on the phone what you want to prioritize. Um, when you're thinking about image analysis, whether it's specifically with napari-ndev or just kind of general goals or gaps that you feel like are missing in your knowledge. Very cool. These are interesting results. Um, I think it's really great that generating and using workflows is a priority of people because when I started out in Napari, I thought the Napari Assistant, which we'll talk about, which allows us to do this, was just such a cool feature and kind of really topped anything that I had experienced in ImageJ. It's also good to see that people are interested here in the batch processing side of thing. And I expected more interest in the easy machine learning, so it's kind of good to know that I don't want to spend as much time on that topic. So yeah, thank you all for your responses. All right, so I think I can bump out of that Slido now and instead come back to the presentation here. So I will jump to this web page in a moment, but please feel free to click this link. This will take you to the documentation for the package, or use your phone to get there. I'll wait a few more seconds for people to do this, but you'll be able to read my browser link as well, probably. Okay, all righty. So this is the documentation webpage. I'll just kind of explain it briefly. Here is kind of the general stuff about napari-ndev, an overview. I'm building up the examples right now. I'm happy with the I2K workshop. I hope that works. I admittedly was working on this quite late last night, so it hasn't been fully proofread, so I hope it works. And then for more sort of advanced developers, there's an API that you can look at. So because most people are new to Dapari, if we look at the tutorial setup here, it discusses that we have two options. And I say, for users unfamiliar with using the command line in Python, I would recommend following the instructions to install from UI at beginner setup. So if we go to beginner setup, there are a few GIFs, and I expect some people joining haven't done Napari yet. So while in the background this presentation is going on, it would be great if you downloaded the latest version of Napari by clicking this link, scrolling all the way down to the bottom to assets, and then clicking the file that matches your operating system. So I'm on Windows, for example, so I would download the windows.exe file. Once you're in Napari, which I will open, so this is the version that you would download right now. It put a shortcut on the desktop for me. It'll probably give you security warnings, just bypass all of that. And from the UI, which you can also see in that documentation. Did I actually open Napari? Sometimes Napari takes a second to open, um, but I think I probably didn't open it there. There we go. Okay, so once you're in here, oh, I did. See, it takes a second sometimes to initialize. We can go to install, uninstall plugins. Then you can search for example, neural dev. It will normally show up down here, even if you have to refresh it. Okay, so right now it's going with the name napari-ndev. Sometimes it picks a different one. And you'll normally have you want to pick the PyPI source in any version, just pick the most recent version. So I will be working with the same setup that you have, and you can see how to do that in these GIFs. I hope that you find them helpful. So just kind of have that downloading in the background. Otherwise, the only other thing you need to set up, which I wouldn't worry right now, is there's 3 download links for the images that we'll use. They're relatively small download links, so you can just download them at the time you click the link. It will just initialize a download of a zip file in your browser. Okay. So what is the origin of napari-ndev? Kind of go back to the presentation. It started because generally I was excited about Napari and Python. I don't really know why. It just seemed a very practical thing for me. I wanted to connect Napari, which with batch processing, that was a priority of mine to start off. I also wanted to, there are so many amazing Python libraries that just haven't been connected yet. And I think in a lot of ways because it's newer, it doesn't have that maturity that something like ImageJ has. And so I want to kind of connect these great tools and even provide a framework for other people trying to also do batch processing. If you want to contribute to this plugin too, I'm happy to have other contributors join as well. I wanted to build a resource specifically for collaborators. And really my goal is that, I'm not kidding, a brand new undergraduate trainee with no experience can do image analysis with this tool. That is the goal. And so we want to create tools that are easy to use and then in the end can produce high quality data. So this is from my Bina poster, and it is, these are just copied from the poster basically. But to sort of give an overview of each widget, the image utilities widget is meant to handle images. It can concatenate weird microscopy files, and we'll always export them right now as OME-TIFF. I hope to add OME-ZAR support in the future. But it also enables us to, for example, this shows a sparse annotation that we would use for machine learning, just quick labels on top. And we can hit, now the widget looks different than when I presented it at Bina, but we can hit the Save Selected Layers button, and then that stores all of that with the metadata compatible, with the file name compatible, so that we kind of like maintain image parity throughout a processing pipeline. It should, and its goal is to produce image or use images of any dimension, so time, multi-channel, and 3-dimensional, or just 2-dimensional, as well as taking RGB images, which I've unfortunately found a few microscopes are stuck in RGB. We can kind of take them away from RGB and reapply them. The Napari assistant here is seen on the left. This is kind of the workflow part that people are quite interested in. And it's a clickable interface. Robert Haas describes it as a calculator. And you can really just kind of, in real time, apply adjustments to your image and process them. You can also go back and look at them. And so that's such a cool tool that we can take that output from that with the Save and Load Workflows button and just apply that in batch on a bunch of images because you have maintained the metadata that is necessary for this. A key feature, I think, of this as well that I haven't highlighted yet is that every time you do something in batch, it will always produce a log file so that you can go in and know every parameter that you did, what images that you did, what workflow file that you used, so that in the future, right, one thing that you sort of lack if you don't have code is that obvious reproducibility that, let's say, a reviewer of your project wants to know what you did, and you did everything in the UI and nothing was saved except for the output, you really wouldn't know how to backtrack. And so one of the goals with these log files is to make it so that you kind of have a resource to allow you to backtrack, allow it to be used for reproducibility. We also have this ability to generate feature sets for machine learning and visualize them. So one thing that you'll note, if you're not familiar with machine learning, it can be overwhelming kind of what's going on with machine learning. But ultimately, when we train a machine learning classifier, we take an image, we define a bunch of features. In other words, we're kind of processing the image in different ways so that maybe we enhance edges or enhance ridges or blobs. And this allows choices to be made that are more than just based on the intensity profile of the image at hand. And so we can visualize that easily to show collaborators that even with just these 16 different features, right, we have a lot of variety in what a decision tree is going to be looking at. Then we can take this feature set. This segmentation was derived after about 20 seconds of effort. That initial sparse annotation that you saw was just run using that classifier that I showed on that feature set I showed on the previous image on one single image, and we produced a decent output. Now, if we spent more time carefully annotating our image, tuning the feature set, and then training this on a batch of images, we can, in about 20 to 30 minutes of work, produce high quality machine learning segmentation of complex cells. Finally, the measure widget, I think, is one of the biggest strengths of napari-ndev, because currently in Python, most outputs of data are really large, long-form data sets. I'll kind of show you a long-form data set. This on the left shows you one specific label per row or object. It also saves that metadata, and here we just want to measure the label ID and maybe its area. In 10 images, this was 65,000 rows already, not something that you can kind of manually do. So we want to summarize that data. Now, in those 10 images, each image is one row. We maintain metadata. So this came from a file that had information about what well and position it was. So we know it's well H9, right? Different images, what the treatments are. And then we can say, okay, there were 15 labels in this image, one label in this image. There are these different means of the information. So it's summarized. You can plot something like that very easily using Excel or Prism. But it also means that if you are more comfortable with coding a data analysis pipeline, you have that kind of raw data set that's very useful for you. So I want to pause here. That's sort of the overview of napari-ndev. I want to know if there's any questions or if setup is going well or poorly for anyone. So I'll just kind of wait a minute. Feel free to chime in. Okay. Well, hopefully that means we're all doing okay. I'm now just going to show you a few examples of what this plugin has powered. So Elizabeth, the graduate student that I mentioned earlier, has been wanting to count the number of cells in a specific region of the brain. This is actually work that I did in graduate school, and I can tell you how much progress that I've made on this compared to what was possible for me in ImageJ. So I was doing this exact same analysis in ImageJ, and I've rebuilt this, well, not rebuilt it, I've designed it entirely different in Napari, and I think that it has a lot more strengths than what I was using previously. In particular, because of that live feedback of building a workflow, the way that you manage ROIs in Napari, I think, has progress that can be made. It does not work like you might be familiar with ImageJ, but that progress is going well. So for example, we want to measure this region in the embryonic brain. This is going to be future somatosensory cortex. But we want to measure these two different nuclei type, and we can see each colored object represents one of the nuclei. And then finally, we can use this workflow to ask what cells actually probably co-occur with this specific transcription factor. So this uses image utilities. These are really challenging images to work with. They come out of the microscope in RGB, like 16-bit RGB images. They're very odd. So we can put that in the right direction, and then we use the workflow to batch process this on hundreds of images. These images are relatively small, maybe about 10 megabytes, and we can process 100 images in less than a minute. So we're really powering up that workflow, which means that if you've designed your pipeline, your next experiment with 100 images, you've already got all of that data in just a few minutes. Here's a different example where now we want to actually annotate multiple different brain regions on a whole slice. I've actually cut off the rest of the slice. And we have really challenging cells to segment. One reason, if you're familiar with doing fluorescence on whole tissue samples, is you can see there's a lot of autofluorescence and background signal with the various um, certain channels due to kind of the meninges around the brain and things like that. And so intensity-based segmentation does not work for something like this. And so if we use machine learning, we can actually do machine learning. These images are somewhere between 100 and 250 megabytes. So it can fit on a typical consumer graphics card. And we can do machine learning to kind of find where these cells are and map them to what regions they are. And this was work done with Chloe. She has some of the most challenging work for this and has really kind of pushed the progress of the development of this plugin. And finally, here's kind of showing you my work where I intend to use machine learning to train classifiers to segment really challenging cell morphologies. This can also be expanded to other things as well. This immuno and analysis was done by an undergraduate, a brand new undergraduate in the lab this summer, Jane Crosby. She is fantastic. And we can see three different markers we have on this image. We also have DAPI, but I just don't show it here. We label phalloidin, which is actin, and then two proteins of interest, NCOA4, which is targets this ferritin protein for degradation, and ferritin, which stores iron. And so we want to know not only how many maybe particles of NCOA4 are inside the cell, but where they're localized. We can map these things because we have really great neuronal morphology. I have not found a tool to do this automatically very well. So that's I really think a strength of this plugin. It's not perfect. Here it looks great, but it's still not perfect by any means. But it does mean that we don't have to manually or semi-automatically label complex cell morphologies. And then the data output from this, right, we can, make inferences about treatment groups, like the cells treated with a chelator have less ferritin at 48 hours in culture, for example, right? So this is really attempting to power data analysis in the end. And then finally, we can also kind of use these tools to segment the morphology, and we're looking at sort of differences in the morphology of these neurons. You can see just how heterogeneous these cells might be. There are hundreds of different looking cells, and kind of appreciating all of that morphology is something that's very important to me. So really powering high throughput, fine-grained analysis. So with that, we'll move on to the interactive tutorial. So the example pipeline, the first thing after we have our Napari set up, we want to download the images for the example pipeline. These are cell painting images. They come from maybe either Beth Samini's group or Ann Carpenter, the Broad Bioimage Benchmark Collection. I think this is a great resource. It's an open source sharing of images, really cool idea. And these images, when I download them from the Bioimage Benchmark Collection, don't have scale information, and the channels are all split. And so one of the goals in the pipeline is we're going to combine those images so that we can have both the scale information and kind of now have a multi-channel image. And then we want to segment various processes of this. So our goal, we kind of investigate the images here. Our goal is to have segmented this is kind of the boundaries of the cell. We have the nuclei. If we subtract the nuclei from the cell boundaries, we get the cytoplasm. And then we can ask a question, for example, like, are these nucleoli segmentations inside the nucleus or inside the cytoplasm? Are mitochondria inside the cytoplasm, inside the nucleus, right? There's a lot of the work that Cell Profiler is doing. So if you're familiar with Cell Profiler, this is a very different take on how to approach that high content screening type analysis or high throughput analysis. And so what this tutorial does is it walks you through using these images. Here, for example, we're going to do batch concatenation of the images, where we select files. We'll select 5 files. So in order to get to plugins, we go to plugins, image utilities. The first time you try to open image utilities, I don't know if this is, I think this is a recent bug with Napari. You'll get this little error, failed to import command. Once you close that down and you try to open it again, it will reopen. I will try to fix this in the future. It doesn't seem like the error is sourced from my plugin. So if you're on a previous version of Napari, let me know if that happens. I just haven't had time to test it yet. So what we'll do is we'll choose our directory, for example, and these are in, let's see, in my file, I have them in this tutorial section. And then in this cell painting folder, what you'll end up having is these images. Oops, where did they go? Oh, that's right. I'm trying to select the directory. There you go. Always one of those things. Everyone always says live tutorials are challenging. So I'm going to select the cell painting folder. Why? Because this is where I want all my outputs to save to. So if I concatenate images, it'll make a concatenated images folder. If I do a workflow in batch, it'll make a folder for you every time. So next, I'm going to select files. So inside this images illuminated corrected, I'm going to select there are 5 channels per image. And we can see that. I apologize if this text is a bit small. I have a monitor that has high resolution. I know that can be challenging on smaller resolution monitors, but there's 5 images here. We can open them. We can see that the metadata, Goost, I don't know if I pronounced that correctly, asked the question, will the recording of this workshop be available somewhere? My Napari is not responding, but I'd like to watch the rest of this tutorial once it does. Yes, so I believe that all presentations from virtual I2K are automatically going to be uploaded to YouTube, so we can thank the organizers for that. I think last year took 2 weeks to a month to be uploaded, but I don't know how long that will be. If you want, you can send me an email. I should have put that somewhere in my presentation, but there's my email if you want to email me, and I'd be happy to email you once it's available. All right, so back to here, and I'll let you kind of work through the tutorial after this. We can select our files, and then we can see here that this metadata information isn't correct. So I want to add the correct metadata information. And so this says, for example, that we want to scale the values to 0.656 microns per pixel. And there we go. So we can put that into the scale. This is in the metadata dropdown. And then also for ease of use, and this is why it's kind of useful, you can just take this little code or snippet of a string here. It's a list of all the different channel names because we know what each channel 1, 2, 3, 4, 5 is. And we can concatenate these images as simple as just hitting batch concatenate. We'll wait a second, and then you can see batch concatenated files are in the directory. And so batch prepping your images is as easy as that. So if we look in here, we now have those images. We now only, instead of having 50 images, we have 10 images. And there we go. If you're not familiar with Napari, you can turn on and off layers like this. We'll be making labels layers throughout this. So this is the labels layer. And if you hit the paintbrush tool, for example, you could draw on labels. You can draw on shapes, for example, and it's a great tool. So let's see what the process will look like for the workflow part of this is you'll choose those images, and you'll specifically want to choose the images that are in the concatenated image folder. We want to save that into a new folder. For example, I'll call this the workshop workflow output. So I made a new folder. You can save it into an old folder. It'll either overwrite images if those names already exist or whatnot. And then doing a batch analysis is as simple as going to where your images are. You can grab this .yaml file, which is available in the download, and it automatically gives the metadata. And the instructions will tell you that the cytomembrane is WGAF phalloidin, the nuclei is H33342, and the nucleoli, I think, is cyto14. Yeah, I think that's right. And then we select the task that we want. So in addition to all of these, we also want to know the nuclei. We hit batch workflow, and there in our 10 images, we're done before I could even finish the sentence on a batch workflow. So if you have thousands of them, you just click the button and you walk away and it'll work for you. Finally, we want to show you the measure widget in this case. And I'm going to select where my labels are. So they're in that workshop workflow output. I can measure multiple labels at once. So I'm going to label the cells. That's that boundary that I showed you, the cytoplasm, and the nuclei. Oh, I also need to make sure, well, I want to choose an image directory. I'm going to add concatenated images. Because, for example, in this instance, maybe I want to measure the intensity of con A, which I think is the ER, and mito tracker deep red, which labels mitochondria. I want to measure their intensity. You know, maybe I can measure the mean intensity of what's under there or the max. I could also measure shape features like how solid is the object, how big is the perimeter. And this will go on to explain how you can also extract metadata from your file names. This is such a cool feature. And we can add a treatment map so that these are from 384 well plates, that you could give it a map of treatment, say, these wells contain this information and this well contains this, so that when you measure this data, all of that is kept. And the beauty of it is, I'll show you how simple it is as well. We'll just take, we'll go down here. We grab this. This is called a dictionary. You don't really need to know the format. This is kind of something that would be maybe a more advanced user would be comfortable making a dictionary because it uses regex and things like that that are really hard for me to even comprehend sometimes. But this is kind of allows that shareability. And let's see, this is the information from the file name and what scene it is, for example. And then this is the information for the plate map. I just made this up. There's actually no treatments mapped to these correctly for this 384 well plate, but we just stick it in there. We update our treatment choices, so we're gonna know it based off of what well it's in, right? Because that's what our identity is. And then we know it's a 384 well plate. We just hit measure, and there we go. We've measured all our images, and we now have a big data set. So what that data set will look like and what I mean by it's very large, this is already, I have no idea, 3,400 rows. But if we group that data, here's those 10 images and all of the various the data that we want from it. So this is kind of our name of our thing. Thank you, Brian. I appreciate it. And I apologize that I never responded to your post on the image forums. So yeah, thank you very much. This lets us know, what site it was. That was just like what position in the well it was. And then here's our treatments mapped. But then we say, okay, we want to count the number of labels. In this case, it makes sense that we have 79 cells, 79 cytolabels, and 79 nucleolabels because they're all the same. But then we can measure the area of it. So it makes sense that the area mean of cell labels is larger than the cytolabels, nucleolabels, and right, they add up. So we can use this also for like quality control check, etc. Grouping your data is fantastic. I love this tool. It is probably the hardest thing to kind of wrap your mind around, especially if you're not used to thinking about big data analysis, but I assure you that it will be such a useful tool for you to use. So I think I would like to try if for people to try to work through this example pipeline tutorial and give feedback. We'll probably come back together around 12 o'clock and I'll show you the other tutorials. Maybe we'll work through them, maybe we won't, but we'll see how this goes. Feel free to ask questions at any time. You could turn your mic on, your camera, whatever, or put it in the chat. So thank you so far for your attention. I love this. Can anyone confirm to me that things are kind of working for you at least? Thank you. I got a response from Emma that it is starting up. That's good. I think maybe just in the background, I'll just kind of be walking through the plugin. Just sort of quietly, so that if anyone does just want to listen, they can. But the priority is for you to kind of ignore me and work on your own. And feel free to interrupt. So I did this image utility step, and I added the channel information. Then we looked at the images and showed the example workflow. One feature that I didn't explain in the workflow widget is a use where if we take our images concatenated images, and we're going to make a new directory for this, and we're going to call this workshop workflow all tasks output. So this is described at some point in one of the tutorials. And I'll select that same workflow file. We'll just put in the correct features here. And then, right, by default, this is labeling all of the end tasks. What we can do, we added before nuclei labels. You have to hold control or command, depending on your operating system. We can also go from just select them all. This will show you everything that is in the plugin. If we do keep original images even, and we do batch workflow on this, and then go to that folder, one of the really cool features, if you want to explain to a different user that you've maybe designed the workflow for, oh, this is a bug that I get sometimes in Napari. If anyone else gets this access violation reading error, please let me know. I would love to know. I've submitted the bug in Napari. There's no error trace. It's something to do with they can't read the image correctly, but it all shows up here on the left and it just doesn't show it in Napari. So if that bug happens to you, I'm very sorry. The I don't know if it's my plugin or not, and it's super intermittent. If it happens, unfortunately, the typical way to deal with it is to restart Napari. Oops. I want the all tasks output. Hopefully when we put that in there, we won't have an issue this time. Oh, there we go. Do you ever get a warning that napari-ndev can't open the image? I also don't know where that's coming from. Um, that just appeared on the most recent version as well, 0.5.4, but also my most recent version of plugin is the first one to contain an image reader. It used to depend on Napari AICS image, but if you're familiar with AICS image IO, it's the best way, I think, to handle most microscopy data in Python. It now has bio-IO support, or bio-IO is kind of the replacement of that, which I think works better. So anyone running into any issues or anything? Okay, so here is all of those tasks that we produced from the workflow, and we add it all on here. It looks like a disaster. It'll load everything in this image. You can see it all like this. But let's say we want to kind of investigate what's going on here. So we can pull this out. We could see everything. So I want to view what's going on. What is nucleoli? What was processed with the nucleoli? So we can hide all unselected layers. And now we just have these. And we can see, for example, let me turn on Cyto 14. So now this is the original image. Here, I'll do it this way. This is the original image of the nucleoli. Then we add a median blur to that. So it's going to look slightly different. Right? There we go. We'll do it. We'll do it translucent, no depth. So there we go. No blur. Now we have that. So we reduce noise. And on top of that, we're going to do a top hat. So what does a top hat do? Kind of reduces the background here. You can see that background is just gone now, right? Where did all of that go? That's a beautiful thing about preprocessing filters. So then when we segment them, it looks like this. Now, when you load these in, they're going to be in an image format, but we can click convert to labels. And now a labels layer can be set to have an opacity, for example. And now we can see each individual label on top of the nucleoli. It did, I think, a great job. It even tries to split neighboring regions. This is called Voronoi-Otsu labeling. I think if we change this to gray, that'll help. So here we can see the end results, but we also want to compare it to the original image, right? So if we want to compare that to the original image, we can turn off the other ones and then go like this. Does it still look good after our pre-processing steps and what we get to with the segmentation? I'm very satisfied with this. This would be great. Yeah. If you're wondering what the file looks like what exactly is this .yaml file? It's instructions here on, please zoom in. It's just starting up. View. Um, Zoom in. Oh, control equal. There we go. It gives all the tasks and stuff. So you could, in theory, if you were comfortable, you could come in here and change the values. What I hope to do is make this possible to do in the widget in the future so that you can actually load in a YAML file and modify these values. But for someone who's experienced, you can update things from here, and that's really great. And then the way that I generated this file isn't available in what you downloaded, but it is available in the GitHub repository. What it looks like to make your own workflow from code is like this, where you basically create a workflow and then you just set the functions and what images it's happening on, etc. All the way through. So that's it. So just these steps were used to create that entire process. That's it. So even from a coding perspective, if this is something that you wanted to do, it's practical to learn. It's a little odd, but it works really well for that reproducibility reason. So that's where that comes from. And yeah, let's wait a moment, see if anyone has any questions. You don't have to be shy. If you want to be shy, that's okay. Okay. We can all take a little stretch break before we move into the next step. Feels good. I hope if you're at these workshops a bunch today that you take some time to move around. In a lot of ways, you know, a lot of people listed that they were new to Napari. A big reason that I'm sitting here giving this Napari workshop is because I watched workshops just like this. So you never know what it'll spark inside uh, of your creativity. And yeah, I never really thought I'd give a workshop either, but people at Bina really encouraged me to do this, and I'm grateful for this opportunity. Okay. So, you know, just want to remind you that really the initial work that we did, well, even I've been chatting for like 10 minutes, but if your workflow just took you 10 minutes to measure 100 images, man, that's a good day if you ask me, because as you might know, doing multiple images is challenging. So because of this, or because of where we're at so far, I want to kind of move on to um, the next, um, the next tutorial. Um, this is going to be a little bit out of order, but specifically because there's an interest in building your own pipeline, I think it would actually be best if we move to tutorial 4. And again, I think you can treat this as kind of like go along. Maybe we'll try like you can go along in the background. You're not going to hurt my feelings. I can't see whether you're paying attention to me either. But I'll just kind of like walk through it myself. If you're able to work through these tutorials on your own, I really want feedback to know if they're useful for you or if there are parts that are confusing. Because I think after I was kind of creating these, I was like, I think these tutorials are the main way that I want people to learn besides watching something like a workshop. I intend to, as I kind of refine this, to make it a um also some YouTube videos that are shorter and more to the point as well. All right. So for the next tutorial, I really should have put a download link on this page. We want to download from the tutorial setup these neural progenitor images. And what those look like are these images. These are already concatenated, so you can just kind of use their data. The goal of this tutorial, of the build your own pipeline tutorial, is for you to kind of figure out how to generate this workflow yourself. So if we load in one of these images, I'm going to go to the neural progenitors data set. You'll just have to unzip that file. You should have concatenated images. I'm just going to open that first one in the link. Here you can see something goofy, as I mentioned, right? It duplicates the data into this channel. That's kind of all right for our purposes, but here's sort of the difference in them. And our goal is to segment these images so that each nucleus has its own label. These are transcription factors in wide field fluorescent microscope images. So we loaded in an image, and one mistake that I made was I didn't load it in with the image utilities widget. However, a really neat feature of this plugin is if we go to metadata, we can actually select a layer and say update metadata from selected layer, and it actually is able to read in the scale and the names of those channels. So we don't technically have to upload it from here, but that was just easier to explain for documentation's sake. And I think it's a better thing to be used to getting your image from here. So, you know, we can select the next image, open the file, and there it is. Another feature of this is we can also hit select next, and now it actually just picks the next file in the folder, and we can open that folder. So this actually really speeds up annotation, because if what we're doing is, you know, we want to make sure we save this into the proper directory. So I'm gonna make sure I set that up. I often forget to choose the save directory, and then the data will always be saved into the default of where Napari is. So it's usually in, like, on a Windows computer, it'd be like C users Tim Monko, and it's just in the base, and it's hard to find. So that's why I would recommend trying to prioritize remembering this. But let's say we pick that first file. And what we want to do is add a region of interest, because we want to count the number of cells in that region of interest. First thing I'm going to do is go to View, Scale Bar. Thank you, Herbert. And we can click Scale Bar Visible, and now it appears. And this is because we have that properly scaled metadata. Next, we're going to draw on a shape using a rectangular ROI. And, you know, this is probably appropriate. Now, one unfortunate thing is a recent update to Napari kind of broke shapes a little bit. But when I reported this bug last night, actually someone already responded. I think they're in Europe. And they said that this is currently fixed on main. So the next release of Napari shouldn't have this issue. And that issue is that once you rotate an object and then try to move it into place, sometimes it's really hard to select it. Like you can see here, I got it deselected and it's not moving. If that happens, you can try to re-rotate it and move it. If that doesn't work, just make it a little longer and you'll be able to move it again. It's really irritating, but again, it'll be fixed in the next update of Napari. If you downgrade to Napari 0.5.3, this bug is not present. So, you know, if you're currently wanting to do this, it's a little buggy, work around that. If you're watching this workshop in the future, just use the most recent version of Napari and it shouldn't be an issue. Now for me, I want to include every cells up into, this is called the peel surface. I don't want to do that because some cells, you know, as you can see here, some of these nuclei go up a little bit farther into the cell. And there's our thing. So then if I hit save selected layers, it's going to save whatever you have highlighted, and it'll intelligently look at the type of the layer and then save it based on that. So here we go, saved shapes. So if we look in our folder now, we will now have a thing called shapes. And this, if we drag it on, looks just like a weird black box. But if we convert to labels, now we have that label image. It basically serves as our region of interest. And that label has a value. If we look down on the bottom of Napari, I wish I could move my mouse, but down here in this area, it'll show you the value of that on this label. This is 1. Everything not labeled has a value of 0. So that's how you can kind of map where a cell is. You know what region it's in based off of its identity. If you want to reproduce, like let's say you have a unique ROI. You've got an ROI like this. The only way right now to kind of save ROIs in Napari natively is to go to Save Selected Layers. And then as you can see here, it saves it as a .csv. If you're familiar with a .csv, it's like an Excel file. It's just a spreadsheet. And I'm just going to quickly save this to my desktop to make it easy. But if we look at, why did I not save it? Save selected layers. Oh, that's saved in the OneDrive version. Anyways, I'll put it actually in the neural progenitors thing. Shapes. Oops, just right in here into the base, shapes.csv. If I drag, and I'm partly teaching this because this is potentially confusing. If I drag shapes.csv on and I load in those shapes, you'll see that they're the wrong scale. And this is because right now, the CSV file just gives you basically the vertexes of that polygon and where they're at. So it has no information on scale. We can't do anything about that. It's unfortunate. But this plugin is designed to actually allow you to do this. So if we delete the shapes layer now, this is the one we loaded, it's the wrong scale, but we have the scale information from other images. So we can just hit scale layers, and now it scales those regions of interest to the correct size, and then we can move them into the position that we need to. So if you have kind of complex ROIs or you need reproducibility on an exact size, that is the workflow that I would use. Okay, so we have our region of interest. To show you how quickly we can annotate images, we can just hit, so we can, well, we want to bring back in this shapes layer. And then we're going to rescale it just to show you kind of how that reproducibility works. I'm going to, in the shapes layer, I'm going to select this one. I don't need this ROI. If you see that bug, list index out of range, local variable res, again, that's going to be fixed in the next version of Napari. So they just made some changes to the shapes layer that are a little funny right now. And so we're going to just, delete these. We can open our next image. So I already hit select next. I'm going to hit open files. And now this is, oh, that's the same image. Just select next. There we go. We're going to delete these, open files. So selected the next. Now we can pull the shape on top and we can rotate this shape into position. So I'm going to do that. Then I'm going to move it. It's a bit long, so I'm going to shorten it. And then I'm going to hit save selected layers. Bam, it's done. Delete, select next, open files, move this on top, move it into position, save selected layers, select next, open files. We don't even have to even delete those layers. It'll fill up your memory eventually and it'll start to bog down a part. But if your images are small, it's okay. There we go. So we're very quickly able to annotate our images. So cool. We now have a folder of our 4 shapes, and we've got our folder of our 4 images. Wonderful. They all will match. So now we want to generate a pipeline for this. So that example that I showed you about how to do this is also the same logic of how you would generate sparse machine learning classifiers. You would just hit save selected layers. It's going to save those labels that you can use them later. Okay. So let's talk about how to train or how to work on an Napari workflow. So if we go to plugins, assistant, these are kind of equivalent. They're just redundant right now. Here's our calculator interface. I want to just focus on this one channel. Personally, I always like when things are gray, and I'm going to think about what I want to do. When I look at it, the kind of the assistant widget here kind of goes in a logical order. We've got the ability to, we can say, let's remove noise. I personally, Gaussian blur is technically faster, but I always like using a median filter because it preserves edges. And so we can actually just sit here and change the difference between them. We can see, okay, this edge between these two nuclei, which we want, is kind of lost a little bit compared to this. But both work, depends on what you're doing. I'm going to remove the background, for example. This is that secret weapon, the top hat sphere. I love it. It works well because your shapes are spheres. This is G, so anything that is from PyClesperanto or PyClesperanto Assistant, if we like look at the list, those are accelerated by your graphics card or your GPU. Anything that's, for example, NSB-ATWM or Napari segment blobs and things with membranes, and or SimpleITK is run on your CPU. But the cool thing about these GPU accelerated ones is I believe if you don't have a GPU, it'll run on your CPU, but everything has an integrated GPU at least these days, and this should work well for that. This also means that when we have large images, like not light sheet large, but 200 megabytes or a gigabyte image, if it can fit into your graphics card memory, then these work fantastic. So it can really accelerate your processing. We had a pipeline for 100 megabyte images, and it would take about like 5 minutes per image on the CPU, and then it took I think like 15 seconds on the GPU. It was a lot of steps, but obviously that's the difference between days and hours for a high throughput workflow. So then finally, we want to do something like, I don't know, we want to click the label button. The secret weapon of PyClesperanto is Voronoi Otsu labeling. I encourage you to learn more. I have a link in the documentation for it. It's in one of the hints. And if we change these are like Gaussian blurs, basically. We change them, we can see it does a really great job of segmenting cells. And there we go. So it's as simple as that. If I just have one thing to do, maybe what I want to do is like say I don't want to count cells that are like, what is this? Well, that's 9 pixels, but let's say I don't want to count something that's like this small, 6 pixels. I could process the labels then and say remove, let's see where, exclude small labels, and then we can pick like I think it's named wrong, but you pick the size you want. So 10, and you'll see now that, oops, yep, anything smaller than 10 pixels is gone. Let's see, let's try 5. That brings some back. Let's see, 1, etc. So, this and then this, that's kind of the difference there. And actually, I think that might be working a little odd right now. They're recently kind of updating oh yeah, oh, it might be doing it to scale. That's a new feature. Yeah. All right, I'll talk about that. They've been converting PyClesperanto prototype version to PyClesperanto full version, and this exclude small labels is kind of a new function in that realm. So, yeah. So let's just say we're happy with these results here. We like this image. We don't, but yeah. So we'll be comfortable with this. Actually, even undoing and redoing things, we can go back. We can say, oh, well, what do these results look like if I increase the median sphere, right? And then it's updating this whole process live for you. It's so cool. I love this about the workflow. So it allows you to go back and forth. If you want to change this to something different, like top hat box, go for it. Now it's using a different algorithm. You want to see something completely different? Try a bottom hat, right? That's going to make your image look really goofy. So not what you want, but at least you can like in real time interact. I think it makes image analysis quite fun. It's like a little game, a little puzzle. When we're done, we can say export workflow to file. And I'm just going to stick that in the folder that we've been working on. So I'm just going to call this workshop zoom.yaml. There we go. We've got our file. And just like before, all you have to do is go to the workflow widget, right? You just spent the time designing that live. You set your images. right? We've got our concatenated images. We're going to make a new directory in here. We're just going to call it workflow output. And then you'll see kind of one of the unique features here. We've got napari-ndev. Oops. Let's go here. We've got that workshop zoom file.yaml. Here we only required one image. It knew that we were looking at this, specifically looking at this TBR2 image. So it lets us know what that root is. We can select our TBR2 file. And then it currently just selects that end task, result of Voronoi Osu labeling. So there you go. That's as simple as that. You designed it, hopefully on your own, and you made a bash workflow. It's that simple and just so usable like that. Do we have any questions? All right. So with the last few minutes we have, I thank you just for your attention so far. With the last few minutes, I will kind of talk about, I don't think I'm sort of missed anything in this tutorial. Oh, right. If you wanted to, measure this kind of encourages you to think about how would I measure this region of interest. Right? It's a label image. We know it has a value of 1 if it's in the cell, and then you can group your data so that ultimately, do I have that in the tutorial? No, I don't. You can, get your data output grouped by whether or not it's in the region of interest or not. Be happy to talk to you more outside of the Zoom if that's something that's really interesting to you. It's definitely commonly done in embryonic brain development, which is what I'm super familiar with. And I've used this for someone, example, we were doing lung tissue and stuff like that. So I don't know, it could be relevant to your field, especially if you do slice work. Here's kind of like, there's hints in here if you're trying to design your own. I think this is kind of a nifty feature. So yeah, so we'll go back to easy machine learning. I want to show you this. This is kind of the least developed plugin. So if you or tutorials, if you go through this and it's confusing, please let me know. This is trained using what are called accelerated pixel and object classifiers. You can see here kind of what the goal of this is. You take what's called a sparse annotation right here. The blue is labeling cells and orange is labeling background or something. And you try to segment the objects. This is called pixel classification. This is where you just give something in the image a class. So object classification segmentation is probably the most common use I would say that I've seen so far. You can also later, I'm adding this to the plugin, you can classify objects. So like these are labeling, this is just the classic blobs.tiff. These are labeling dividing cells, the screen one are labeling single cells, and then you can see, oh, we've got dividing cells and we've got single cells or elongated. And it's really neat. I currently use this in my own work to identify in like whole well images of like the nuclei stain, whether that's DAPI or hoaxed or something, to count the number of dead and alive cells, because we can quite easily segment DAPI, and then dead and live cells are very different. And so we can use this object classification to screen really quickly how many cells we have that are alive. It also means for like my single cell neuron stuff that I can actually get rid of cells that maybe have multiple nuclei or have dead nuclei, because we don't want to quantify those when we're trying to consider live cells. So you always kind of have to be thinking about what analysis you want to do. So this has some great examples in it. The documentation, and Robert Haas, which is what a lot of this plugin is built upon, his work. He just has so many example documentations. Like seriously, the bioimage, and I always search it, bioimage analysis notebooks from Hasleinhof. That's just, I think he's from Germany. I don't know if that means something in German, but this is Robert Haas's work and a few other contributors. This is such a great tool to learn about Python image analysis, also just image analysis in general. Um, Super cool. It's kind of a lot of where this all started. Okay, so let's go back to here and uh, our tutorial. Here we go. Easy machine learning. So we talked earlier, let's load in one of these neuron images. Okay, so one of the wonky things, I'll show you something. This is expected behavior. This is just how the plugin works. Let's say I load in one of my primary neuron images, right? It has absolutely nothing to do with that workflow I did. That workflow is in memory right now in Napari. So when I load in this image, it's going to either crash Napari or it's going to try to do that workflow on the file that I give it. Yeah, I'll wait a second to see if it uncrashes Napari. If your Napari ever does this, this is because it's thinking. Um, yeah, it seems like oh no, it did do it. Okay, so you could see this is why it was chaos. So it tried to run the first channel that opened. So it tried to do that process that we did on that first channel, and ultimately that resulted in wherever it is. Uh, uh, Failed to read memory buffer. Yeah, this really broke it. If you look on the left, it tried to make a label image a median sphere. So that's going to break things. So if you're ever done with your workflow, my advice, close Napari, reopen Napari, and start again, because it's always going to be kind of doing something funny every time you load in an image. It's a quirk. Maybe I'll try to assist that package so that no longer happens. But here we go. Back in business. All right. So let's load in. Yeah, failed import command. Oh, I wish it didn't do that. Anyways, select files. We're going to go to primary neurons. We're going to take these are from multi-scene CZI files. So from a Zeiss microscope. Great management of metadata. I didn't have to worry about that. And I just pressed this extract and save scenes button. And if you do that, it'll just extract them all. But you could also, like, let's say you want to screen a few images, you could be like, I want scene or image, whatever, 10, 50, and 100. And then you can hit extract and save scenes and it'll do that for you from multi-scene data. Really neat feature. Here we can see though that this has the correct metadata. We're going to open this file and there we go. We've got pretty little neurons. I hope these are beautiful to you. These are wide field epifluorescent images again. And here we've got this is nuclear coatic factor 4, not a very bright signal in this particular cell. We've got ferritin, we've got phalloidin, and then we also have the nuclei. So let's use machine learning quick in our last few minutes to show you how cool and easy this is. So let's just visualize this. If you're really just wanting to do one channel, put it on gray. Your eyes will always thank you. It's easier. You can change contrast here if you need to, etc. We're just going to throw some labels on here. So as a convention, it's helpful to label your background with 0. So I like to go around the edge. I'm just kind of trying to tell you the logic of how I do something like this. Because as you can see, on the edge of this, right, there's a blur, but that's not the signal that we want. And we can help train the classifier. We can also just do some stuff out here. There's nothing really out there, but you're just saying, if you're making a decision, right, you want to know what label 1 is, just look in those areas, make decisions based on that. And this is where it's you look at this and it's okay, there's not much signal here, but you're telling me you want to segment the soma with this? Okay, well, let's do some soma. Let's do the other soma. We're just doing sparse annotation. We can do thicker branches. We're trying to get a variety of what we have here. We can change the brush to be smaller. So we go down thin elements, right? I'm being brisk here, but this is the idea. You don't need to be perfect. With deep learning, as you might know, if you've ever explored using deep learning, you have to be perfect to do this. And it's tough. So we want to get a variety of stuff. These are dimmer areas of phalloidin. This is the growth cone of the neuron. Cool, we want to label this stuff. You could even be like, do I want to try to get this? Okay, that's really thin. Maybe I'll draw it here. I don't know if I'd really recommend doing that. But hey, if that's the feature that you want to extract, go for it. So from here, how can we do machine learning? Plugins, APOC widget. Now we have this cool widget. It'll open really wide if you have images because it's trying to like fit them all in this box. I think it's bothersome. But what we want to do is create a classifier file. So I'm just going to call this, oh, we have to make it like a text document. I'm going to call this workshop classifier.cl. We're going to delete the txt. It's going to say, if you change a file name extension, the file might become unusable. You want to change it. So here we go. Workshop classifier.cl. I think that's the one I just made. Yep, has no data in it. So let's say I want to just keep this super simple. I want to test, what does it look like if I just say the objects are 3 to 8 pixels, or actually 1 to 5. Why not that, right? Because we could look at this as more like 15 across. I can assure you that some smaller radii would be helpful. Neurons are goofy. We could talk more about how to do neuron morphology if you want in an email. Um, I think this is one of the most challenging things to do. We use this on axons. We've used this on kind of, it looks like cheesecloth to me, like lung tissue. We've used it on retinas. Lots of different kind of sites from everything from like a sort of a mesoscale structure, like veins going through an eye, to these small scale structures, because everything is just like, what is the scale you're looking at relative to the number of pixels it has? And this has like a width of like 8 pixels. So the channel that has phalloidin is 488. And so when we select a feature string here, it's just going to say, okay, we're going to do a Laplacian of Gaussian in the box shape of a Gaussian blur of width 2, right? That's what all this is. This is probably nonsense sometimes to you maybe, but it's as simple as train classifier on selected layers using label. You'll get a little pop-up here. It says cannot continue training. So it's saying this is a new classifier. And then we just say predict on this image. And there you go. I mean, this is really good. If you've ever tried to segment complex cells, this looks really great, and we barely put any effort into it. Now, one of the issues that if we look at this is it's like way too big, right? This is not how big we want our objects to be. This could be because we labeled too much of the soma, for example, or that we didn't label enough fine particles, or that our object size is too big. So, we can try again. We're going to turn off continue training so that it overwrites it. And then we're going to say predict using classifiers on select labels. All right, that's a little tighter, but I still don't really like this. So what happens if we add another channel, for example? What if I train it now? Oh my, we got really wide there. There we go. That is something that I will fix. I know why this is happening. I recently learned how to fix this, so I'll do that. But now if we look, great, we've got all these labels. Oh, we're getting a much more fine, sorry, it's so big. We're getting a much more fine apparatus, right? So we can tune this up. But how do we really know what these features are that we're giving a decision tree? Um, we can go to custom feature set, right? And we can pick those features again. Let's say we pick 1 to 5 pixels. It automatically generates the values in here. And we can say, for example, we're just on let's see, we want to select the 488 channel again, right? Because it has phalloidin. And now we can apply that to the selected image. And there we go. Super neat. We now have our image with all of our different features. So here's the original. That's a Gaussian blur of 1, 2, 3, 4, 5. Difference of Gaussian 1, right? Completely different features. Off, 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 right? And now as it goes bigger, it changes, right? Now we've got almost ridges being enhanced. We've got a different one. This one does ridges well or edges well, etc. You can also, let's just turn everything back on. Show all. You can look at it like this, like I show in the tutorial. Another thing that you can do is to make your own feature set. So for example, I know that ridges work well. Oh, it's a bummer. I just deleted the labels, but I know ridges work well with something called a Hessian matrix. That's kind of what a neuron is. So you could be like 1, 2, 3, 1, 2, 3, generate feature string. You could try that on this image. And there we go. If we zoom in now, we can see that this completely different feature set, right? So this could be useful for your case. So you kind of have to play around. And then what's really neat, if we load in a classifier, for example, it'll give us a little pop-up here. that tells us kind of what was most useful. So for example, a Gaussian blur of 2 was used heavily. It was, this is out of 1, was 54% of the decision, where these other ones weren't used. So you can kind of like use this as feedback to tune your machine learning classifier. From here, batch processing is as simple as you have that classifier selected, you choose your directory and channels like you would in the measure widget, and you go from there. Yeah, I'm seeing that it is in my time. It's been an hour and a half, basically. There's 2 minutes left, but I just wanted to show you how easy machine learning um, can be and practical it can be. Um, wow, I just kind of sorry, a little bit of a steamroll at the end, but thank you all for being here. I'm really grateful that you've taken time out of your busy life to learn more about this. So I'll be sitting here if people want to ask questions. I know you might want to move on to your next topic. But yeah, thank you so much. I feel honored that you were here.